Hello friends, today we are going to read neural control and coordination. As you know, the functions of organ system in our body must be coordinated to maintain homeostasis. Coordination is the process through which two or more organs interact and complement the functions of one another. For example, when we do physical exercise, the energy demand is increased for maintaining an increased muscular activity. The supply of oxygen is also increased. The increased supply of oxygen necessitates an increase in the rate of respiration, heartbeat, and increased blood flow via blood vessels. When physical exercise is stopped, the activities of nerves, lungs, heart, and kidney gradually return to their normal conditions. Thus, the functions of muscles, lungs, heart, blood vessels, kidney, and other organs are coordinated while performing physical exercise. In our body, the neural system and the endocrine system jointly coordinate and integrate all the activities of the organs so that they function in a synchronized fashion. The neural system provides an organized network of point-to-point connections for quick coordination. The endocrine system provides chemical integration through hormones. In this chapter, you will learn about the neural system of human mechanism of neural coordination, like transmission of nerve impulse, impulse conduction across synapse, and the physiology of reflex action. Neural system. The neural system of all animals is composed of highly specialized cells called neurons, which can detect, receive, and transmit different kinds of stimuli. The neural organization is very simple in lower invertebrates. For example, in Hydra, it is composed of network of neurons. The neural system is better organized in insects, where a brain present along the number of ganglia and neural tissues. The vertebrates have a more developed neural system. Human neural system. The human neural system is divided into two parts: the central neural system CNS and the peripheral neural system PNS. The CNS includes the brain and the spinal cord, and it is the site of information processing and control. The PNS comprises of all the nerves of the body associated with the CNS, brain, and spinal cord. The nerve fiber of PNS are of two types: afferent fibers and efferent fibers. The afferent nerve fiber transmit impulses from tissue organs to the CNS, and the efferent fiber transmit regulatory impulses from the CNS to the concerned peripheral tissues organs. The PNS is divided into two divisions called somatic neural system and autonomic neural system. The somatic neural system delays impulses from CNS to skeletal muscle, while the autonomic neural system transmits impulses from the CNS to involuntary organs and smooth muscles of the body. The autonomic neural system is further classified into sympathetic neural system and parasympathetic neural system. Neurons as a structural and functional unit of neural system. A neuron is a microscopic structure composed of three major parts, namely cell body, dendrites, and axon. The cell body contains cytoplasm with typical cell organelles and certain granular bodies called nasal granules. Short fiber which branch repeatedly and project out of the body cell also contains nasal granules and are called dendrites. These fibers transmit impulses towards the cell body. The axon is a long fiber, the distal end of which is branched. Each branch terminates as a bulb-like structure called synaptic knob, which poses synaptic vesicles containing chemicals called neurotransmitters. The axon transmits nerve impulses away from the cell body to a synapse or to a neural muscular junction. Based on the number of axon and dendrites, the neurons are divided into three types, that is multipolar with one axon and two or more dendrites found in the cerebral cortex, bipolar with one axon and one dendrite found in retina of eye, and unipolar cell body with one axon only, found usually in the embryonic stage. These are two types of axons, namely myelinated and non-myelinated. The myelinated nerve fibers are enveloped with Schwann cell, which forms a myelin sheath around the axon. The gap between two adjacent myelin sheaths are called node of Ranvier. Myelinated nerve fibers are found in spinal and cranial nerves. Unmyelinated nerve fibers is enclosed by Schwann cell that does not form a myelin sheath around the axon and is commonly found in autonomous and the somatic neural systems. Generation and conduction of nerve impulses. Neurons are excitable cells because their membrane are in a polarized state. Do you know why the membrane of a neuron is polarized? Different types of ion channels are present on a neural membrane. These ion channels are selectively permeable to different ions. When a neuron is not conducting an impulse, that is resting, the axonal membrane is comparatively more permeable to potassium ion K+, and nearly impermeable to sodium ions Na+. Similarly, the membrane is impermeable to negatively charged protein present in the exoplasm. Consequently, the exoplasm inside the axon contains high concentration of potassium ion and negatively charged proteins and low concentration of sodium ion. In contrast, the fluid outside the axon contains a low concentration of potassium ion and a high concentration of sodium ion, and thus form a concentration gradient. These ionic gradients across the resting membrane are maintained by active transport of ions by sodium-potassium pump, which transport 3Na outward and 2K into the cell. As a result, the outer surface of the axonal membrane possesses a positive charge, while the inner surface becomes negatively charged and therefore is polarized. The electrical potential difference across the resting plasma membrane is called as the resting potential. You might be curious to know about the mechanism of generation of nerve impulse and its conduction along an axon. When a stimulus is applied at site, you might be curious to know about the mechanism of generation of nerve impulse and its conduction along an axon. When a stimulus is applied at site point A on the polarized membrane, the site A becomes freely permeable to sodium ion. This leads freely permeable to sodium ion. This leads to rapid influx of sodium ion, followed by the reversal of the polarity at that site. That is, the outer surface of the membrane becomes negatively charged and the inner sides become positively charged. The polarity of membrane at the site A is thus reversed and hence depolarized. The electrical potential difference across the plasma membrane at the site A is called axon potential, which is in fact termed as nerve impulse. At sites immediately ahead the axon, membrane has a positive charge on the outer side and a negative charge on its inner surface. As a result, the current flows on the inner surface from site A to site B. On the outer surface, current flows from site B to site A to complete the circuit of current flow. Hence, the polarity at the site is reversed and axon potential is generated at site B. Thus, the impulse axon potential generated at site A arrives at site B. The sequence is repeated along the length of the axon and consequently the impulse is conducted. The rise in the stimulus-induced permeability to sodium ion is extremely short-lived. It is quickly followed by a rise in permeability to potassium ion. Within a fraction of a second, potassium diffuses outside of the membrane and restores the resting potential of the membrane at site of excitation, and the fibers becomes one more responsive to further stimulation. Transmission of impulse. A nerve impulse is transmitted from one neuron to another through junctions called synapses. A synapse is formed by the membrane of a presynaptic neuron and a postsynaptic neuron, which may or may not be separated by a gap called synaptic cleft. There are two types of synapses, namely electrical synapse and a chemical synapse. An electrical synapse, the membrane of pre- and postsynaptic neurons are in a very close proximity. Electrical currents can directly flow from one neuron into the other across these synapses. Transmission of an impulse across electrical synapses is very similar to impulse conduction along a single axon. Impulse transmission across a synapse is always faster than the across a chemical synapse. Electrical synapses are rare in our system. At a chemical synapse, the membrane of the pre- and postsynaptic neurons are separated by a fluid-filled space called synaptic cleft. Do you know how the presynaptic neuron transmits an action potential? Do you know how the presynaptic neuron transmits an impulse action potential across the synaptic cleft to the postsynaptic neuron? Chemicals called neurotransmitters are involved in transmission of impulse at dead synapses. The axon terminals contains vesicles filled with these neurotransmitters. When an impulse action potential arrives at the axon terminal, it stimulates the movement of the synaptic vesicles towards the membrane where they fuse with the plasma membrane and release their neurotransmitters in the synaptic cleft. The released neurotransmitters bind to their specific receptors present on the postsynaptic membrane. Receptors are present on the postsynaptic membrane. This binding opens ion channels, allowing the entry of ions which can generate a new action potential in the postsynaptic neuron. The new potential developed may be either excitatory or inhibitory. Central neural system. The brain is the central information processing organ of our body and acts as command and control system. It controls the voluntary movements, balance of the body, functions of vital involuntary organs, lungs, heart, kidneys, etc., thermoregulation, hunger and thirst, circadian 24-hour rhythm of our body, activities of several endocrine glands, and human behavior. It is also the site for processing of vision, hearing, speech, memory, intelligence, emotions, and thoughts. The human brain is well developed and protected by the skull. Inside the skull, the brain is covered by cranial meninges consisting of an outer layer called dura mater, a very thin middle layer called arachnoid, and an inner layer which is in contact with brain tissue called pia mater. The brain can be divided into three major parts: forebrain, midbrain, and hindbrain. Forebrain contains thalamus, hypothalamus, cerebrum. Midbrain contains a lot of things. Hindbrain contains pons, medulla, cerebellum. Forebrain. The forebrain consists of cerebrum, thalamus, and hypothalamus. Cerebrum forms the major part of the human brain system. A deep cleft divides the cerebrum longitudinally into two halves, which are termed as left and right cerebral hemispheres. The hemispheres are connected by a tract of nerve fibers called corpus callosum. The layer of cell which covers the cerebral hemisphere is called cerebral cortex and is thrown into prominent folds. The cerebral cortex is referred to as gray matter due to its grayish appearance. Neuron cell bodies are concentrated here, giving the color. The cerebral cortex contains motor areas, sensory areas, and large regions that are neither clearly sensory nor motor in function. These regions, called as associate areas, are responsible for complex functions like intersensory associations, memory, and communication. Fibers of tracts are covered with myelin sheath, which constitute the inner part of cerebral hemisphere. They give an opaque white appearance to the layer and hence is called the white matter. The cerebrum wraps around the structure called thalamus, which is a major coordinating center for sensory and motor signaling. Another very important part of brain called hypothalamus lies at the base of thalamus. The hypothalamus contains a number of centers which control body temperature, urge for eating and drinking. It also contains several groups of neurosecretory cells which secrete hormone called hypothalamic hormone. The inner parts of cerebral hemisphere and a group of associated deep structure called amygdala, hippocampus, etc. forms a complex structure called the limbic lobe or limbic system. Along with the hypothalamus, it is involved in the regulation of sexual behavior, expression of emotional reactions, that is excitement, pleasure, rage, and fear and motivation. Now midbrain. The midbrain is located between the thalamus and hypothalamus of forebrain and pons of the hindbrain. A canal called cerebral aqueduct passes through the midbrain. The dorsal portion of midbrain consists mainly of four round swelling slopes called corpora quadrigemina. Midbrain and hindbrain forms the brainstem, except cerebellum. Brainstem contains pons, medulla, and midbrain. Hindbrain. The hindbrain comprises pons, cerebellum, and medulla, also called medulla oblongata. Pons consist of fiber tracts that interconnect different regions of the brain. Cerebellum have very convoluted surface in order to provide the additional space for many more neurons. The medulla of the brain is connected to the spinal cord. The medulla contains centers which control respiration, cardiovascular reflexes, and gastric secretions. Reflex action and a reflex arc. You must have experienced a sudden withdrawal of a body part which comes in contact with objects that are extremely hot, cold, pointed, or animals that are scary or poisonous. The entire process of response to a peripheral nervous stimulation that occurs involuntarily without conscious effort or thought and requires the involvement of part of the central nervous system is called a reflex action. The reflex pathway comprises at least one afferent neuron receptor and one efferent effector or excitated neuron, approximately arranged in a series. The afferent neuron receives signal from a sense organ and transmits impulse via a dorsal nerve root into the CNS at the level of spinal cord. The efferent neuron then carries signals from CNS to the effector. The stimulus and response thus forms a reflex arc and shown below in the knee-jerk reflex. You should carefully study figure 21.5 to understand the mechanism of knee-jerk reflex. Sensory reception and processing. Have you ever thought how do you feel the climate change in the environment? How do you see an object and its color? How do you hear a sound? Sense organs detect all types of changes in the environment and send appropriate signal to the CNS, where all the inputs are processed and analyzed. Signals are then sent to different parts, centers of the brain. This is how you can sense changes in the environment. In the following sections, you will be introduced to the structure and functioning of eye sensory organ for vision and the ear sensory organ for hearing. Eye. One-paired eyes are located in the sockets of a skull called orbits. A brief account of the structure and functions of human eye is given in the following sections. Parts of an eye. The adult human eye is nearly a spherical structure. The wall of eyeball is composed of 3 layers. The external layer is composed of dense connective tissue and is called the sclera. The anterior portion of this layer is called the cornea. The middle layer, choroid, contains many blood vessels and look bluish in color. The choroid layer is thin over the posterior two-thirds of the eyeball, but it becomes thick in the anterior part to form the capillary body. The ciliary body itself continues forward to form a pigmented and opaque structure called iris, which is visible color portion of the eye. The eyeball contains a transparent crystalline lens which is held in place by ligaments attached to the ciliary body. In front of the lens, the aperture surrounded by the iris is called the pupil. The diameter of pupil is regulated by the muscle fiber of iris. The inner layer is retina and it contains 3 layers of neural cells from inside to outside: ganglion cells, bipolar cells, and photoreceptor cells. There are 2 types of photoreceptor cells, namely rods and cones. These cells contain the light-sensitive proteins called the photopigments. These cells contain the light-sensitive proteins called the photopigments. The daylight photopic vision and the color vision are functions of cones, and the twilight scotopic vision is the function of rods. You can learn it by R for rath and R for rods, and C for color and C for cones. The rods contains a purplish red protein called the rhodopsin or visual purple, which contains a derivative of vitamin A. In the human eye, there are 3 types of cones which possess their own characteristic photopigments that respond to red, green, and blue lights. The sensations of different colors are produced by various combinations of these cones and their photopigments. When these cones are stimulated equally, a sensation of white light is produced. The optic nerve leaves the eye and the retinal blood vessels enter it at a point medial to and slightly above the posterior pole of the eyeball. Photoreception cells are not present in that region and hence it is called the blind spot. At the posterior pole of the eye, lateral to the blind spot, there is a yellowish pigmented spot called macula lutea, which with a central pit called the fovea. The fovea is a thinned out portion of the retina where only the cones are densely packed. It is the point where the visual acuity resolution is the greatest. The space between the cornea and the lens is called the aqueous chamber and contains thin watery fluid called aqueous humor. The space between the lens and retina is called the vitreous chamber and is filled with transparent gel called vitreous humor. Mechanism of vision. The light rays in visible blebulin focused on the retina through the cornea and lens generate potentials, impulses in rods and cones. As mentioned earlier, the photosensitive compounds photopigments in the human eye is composed of opsin, a protein, and retinal and aldehyde of vitamin A. Light induces dissociation of retinal from opsin, resulting in changes in the structure of the opsin. This causes membrane permeability change. As a result, potential differences are generated in the photoreceptor cells. This produces signals that generate action potential in the ganglion cell through the bipolar cells. These action potential impulses are transmitted by the optic nerve to the visual cortex area of the brain, where the neural impulses are analyzed and the image formed on the retina is recognized based on early memory and experience. Now the ear. The ear performs two sensory functions, hearing and maintenance of body balance. Anatomically, the ear can be divided into three major sections called outer ear, the middle ear, and the inner ear. The outer ear consists of the pinna and external auditory meatus canal. The pinna collects the vibration in the air which produce sound. The external auditory meatus leads to inward and extended up to tympanic membrane, the eardrum. There are fine hairs and wax-secreting glands in the skin of the pinna and the meatus. The tympanic membrane is composed of connective tissue covered with skin outside and with mucous membrane inside. The middle ear contains three ossicles called malleus, incus, stapes, hammer, and stirrup, which are attached to one another in a chain-like fashion. The malleus is attached to the tympanic membrane and the stapes is attached to oval window of the cochlea. You can learn it by so, S-O, so for step, S for stapes and O for oval window. So SO. MT, malleus and tympanic membrane. The ear ossicle increases the efficiency of transmission of sound waves to the inner ear. An eustachian tube connects the middle ear with the pharynx. The eustachian tube helps in equalizing the pressure on either side of the eardrum. The fluid-filled inner ear called labyrinth consists of two parts, the bony and the membranous labyrinths. The bony labyrinth is a series of channels. Inside these channels lies the membranous labyrinth, which is surrounded by a fluid called perilymph. The membranous labyrinth is filled with fluid called endolymph. The coiled portion of the labyrinth is called cochlea. The membrane constituting cochlea, the resonance, and the basilar divides the surrounding perilymph-filled bony labyrinth into an upper scala vestibuli and a lower scala tympani. The space within cochlea called scala media is filled with endolymph. At the base of the cochlea, the scala vestibuli ends at the oval window while the scala tympani terminates at round window, which opens to the middle ear. The organ of Corti is a structure located on the basilar membrane which contains hair cells that act as auditory receptors. The hair cells are present in a row on the interior side of the organ of Corti. The basal end of the hair cell is in close contact with the afferent nerve fibers. A large number of processes called stereocilia are projected from the apical part of each hair cells. Above the rows of the hair cells is a thin elastic membrane called tectoral membrane. The inner ear also contains a complex system called vestibular apparatus located above the cochlea. The vestibular apparatus is composed of three semicircular canals and the otolith macula is the sensory part of sulcal and utricle. Each semicircular canal lies in a different plane at right angle to each other. The membranous canals are suspended in the perilymph of the bony canal. The base of canal is swollen and is called ampulla, which contains a projecting ridge called crista ampullaris, which has hair cells. The sulcal and utricles contains a projecting ridge called macula. The crista and macula are the specific receptors of the vestibular apparatus responsible for maintenance of balance of the body and the posture. Mechanism of hearing. How does ear convert sound waves into neural impulses which are sensed and processed by the brain, enabling us to recognize a sound? The external ear receives sound waves and directs them to the eardrum. The eardrum vibrates in a response to the sound waves, and these vibrations are transmitted through the ear ossicle, malleus, incus, and stapes to the oval window. The vibrations are passed through the oval window on to the fluid of the cochlea, where they generate waves in the lymphs. The wave in the limbs induce ripples in the basilar membrane. These movements of the basilar membrane bend the hair cells, pressing them against the tectoral membrane. As a result, nerve impulses are generated in the associated afferent neurons. These impulses are transmitted by the afferent fiber via auditory nerves to the auditory cortex of the brain, where the impulses are analyzed and the sound is recognized. Thank you.